So I got cut off a little bit on that last part, but just to review a little. So as the red blood cells pass through the capillary beds in the lungs, oxygen binds into the hemoglobin within the red blood cells. And again, so oxygen that you breathe in, oxygen-rich air, diffuses into the capillaries because they're thinly walled. Then the oxygen binds to the hemoglobin within all those the millions of hemoglobin molecules within all the red blood cells in the bloodstream, and then the blood now is oxygen rich, heading towards the heart, and then the blood will enter in the systemic circulation to deliver that oxygen to the cells and the tissues. So in the systemic circuit now, the hemoglobin will unload, release the oxygen to the cells. So here is another alveolus, right? Air sac in the lungs. Oxygen diffuses across the membrane, the alveolar cell wall. Then that's the blood capillary cell wall. And the oxygen diffuses across readily and rapidly into the oxygen binding to the hemoglobin. This HB means hemoglobin within the red blood cells. Then the red blood cells move off and blood to the heart. This is part of the pulmonary circuit. Then in the heart pumps and the blood now is entering into the systemic circuit, getting to the tissues at the systemic capillary beds. And then there, the oxygen moves in the opposite direction. The oxygen releases from the hemoglobin, and the oxygen diffuses across the capillary wall into the interstitial fluid and is now entering into the cells and the tissues. So the red blood cells are specialized cells for transporting and carrying oxygen. They're biconcave discs, so there's kind of like they're pinched in the middle. They're almost like a donut shape, but the donut hole is still in place. Okay, so biconcave disc. And that shape of that cell really maximizes, increases surface area for ease of diffusion of the gases, gas exchange. The cells, when they're mature and they're floating around in your bloodstream, the red blood cells, they do not have nuclei or other organelles. So they're not synthesizing anything. They're not making proteins or anything. They're not utilizing the oxygen they pick up. They're basically just streamlined cells, highly modified to transport oxygen in the cardiovascular system. So anemia is a condition with abnormally low amounts of hemoglobin and or low red blood cells. And that can lead to low oxygen delivery at our tissues and could lead to kind of a pale coloration to the skin. So when this happens, when you have not enough oxygen in your blood due to not enough red blood cells, the kidneys will sense this and produce a hormone called erythropoietin, EPO. That stimulates the red bone marrow to produce more red blood cells, hopefully to get rid of the problem of anemia and get your red blood cell count up to normal. Okay, so let's look at what some of the other cells do, like the white blood cells. There are immune system cells for defense. So the white blood cells are also called leukocyte, leuko referring to white and cyte for cell. And we have a number of different types of white blood cells or leukocytes that we won't identify in this chapter. But they do contain nuclei and other organelles, so they are full cells in that sense, or true cells in that sense. They can have metabolic processes, they can produce proteins, they can move around, they can be active. They are larger, but less abundant than the red blood cells. However, due to infections, they can temporarily increase their numbers significantly when the body is fighting an infection. So here's an interesting little video of a white blood cell migrating through the blood and tracking down a bacterium bacterial cell to ingest it and destroy it through phagocytosis. So let's talk about the platelets and how they're involved in the blood clotting cascade. So blood has two components to form clots. They have platelets, which are like cell fragments, which we see here, and a protein that's called fibrinogen. And these are in solution in the blood. The platelets are bits of a larger type of cell that's produced in the bone marrow. So pieces of this larger cell form the platelets, and the platelets are floating around in your blood supply, in your bloodstream, in your blood vessels. And then fibrinogen is a protein that is soluble. That means it's dissolved in the plasma. It's floating around in the bloodstream. However, under the appropriate conditions and the appropriate signals of the blood clotting cascade, the soluble protein fibrinogen will become a solid fibrin that forms this patchwork or a meshwork at a site of injury, a cut, for example, and that will trap red blood cells. The fibrin traps the red blood cells to plug the break or the cut, forming a blood clot. So the blood clotting cascade, that's often called, is an elaborate sequence of events that is initiated when there's a tissue damage at a blood vessel to plug the vessel with a clot, and then leading to healing of the damaged blood vessel. But all I need you to know for that is that the platelets and the fibrinogen that becomes a solid fibrin molecule or protein work to form a blood clot whenever we have an injury at a blood vessel, a break in a blood vessel. Hemophilia is this inherited disease that's pretty nasty, and what happens is it can lead to fatal bleeding, even from a minor cut or bruise, because the normal blood clotting cascade is interrupted because there's a genetic mutation in one of or more of the genes that code for the blood clotting factors, which are proteins. So this is highly simplified here, but if one of these proteins is not properly produced because of a genetic mutation, it could lead to the lack of a clot formation, which means even a small cut will bleed profusely, so much so that the person can die from bleeding out. And your textbook mentions that the problem of hemophilia appeared in the royal families in Europe when they would intermarry, and leading to an increase of the likelihood of hemophilia within members of the royal family. So this is Queen Victoria of England and her husband, Albert. He did not have the condition. Queen Victoria was a carrier and passed it on to one of her daughters, Alice, who married Louis, I believe from France. They had a daughter, Alexandra, who married the Tsar of Russia, Tsar Nicholas II of Russia, and passed on the hemophilia disease to one of their sons, Alexis. And what happened in this time frame in Europe is there was a lot of interbreeding between close relatives to maintain power in royal families. So what happened is hemophilia became much more common in the royal families and in the general population because there was too much inbreeding going on, which is one of the biological reasons why you shouldn't marry close relatives like first cousins, besides the normal ick factor. So continuing, too much or too little clotting can be life-threatening. If too much clotting occurs for various regions, an embolus can occur. This is a blood clot that would occur within a blood vessel that dislodges, and now you have a clot floating through the blood vessels, and if the clot gets jammed in the smaller blood vessel, it can form a blockage there. And of course that's not good, because downstream, if you will, of the blockage, there's a lack of delivery of nutrients like oxygen to tissues, and that's not good. So an embolus can block blood flow to the heart and can lead to a heart attack. An embolus can block blood flow to the brain, leading to a stroke. This is a section of a human brain with damage due to a stroke. So these are possibilities due to too much clotting occurring, and why that might happen is problems with the blood clotting cascade, which is a pretty elaborate system to prevent bleeding from a cut. And hemophilia, again, is a problem with too little clotting ability, which in itself could be bad. So donating blood. So excessive bleeding in some diseases can be treated through blood transfusions, which are relatively very safe and a common way of dealing with excess blood loss. However, notice on the bag here it says type O positive, and it's upside down, but O positive. That's a blood type. So the patient must be able to accept O positive blood, and we'll talk about how they determine that based on blood matching. So blood typing is done, blood matching to ensure appropriate transfusions and minimizing any potential cross reactions or negative reactions that can cause clumping or something that's called agglutination of the red blood cells, which can be harmful. Worst case scenario, it can even be fatal. So if we look here at the blood grouping, blood types, you've heard of probably blood type A or blood type B or blood type AB or blood type R. What's missing here is what's called the RH factor or the negative factor. There's blood type A positive and A negative. There's B positive and B negative. There's AB positive and AB negative. And then there's blood type O positive and O negative. But we'll just simplify that and just say A, B, AB, or blood type O. You are one of these blood types based on your genetics, based on your blood genotypes that you inherited from your parents. So if you're blood type A, where's my cursor here? If you're blood type A, your red blood cells have a surface molecule A. If you're blood type B, your red blood cells have a surface molecule type called B, or carbohydrate B. If you're blood type AB, you have both. Go back and look at our genetics chapter. That's an example of codominance. If you're blood type O, you have none, or you do not have surface molecule A or B. Okay? So in your bloodstream, you also have antibodies to protect you from the red blood cell surface molecule that you do not have. For example, blood type A, they have surface molecule A on their red blood cells, so they produce an antibody B to protect them from something they don't have, like to protect them from a foreign red blood cell molecule, or a red blood cell with surface molecule B. All right? If you're blood type B, your red blood cells have surface molecule B, and in your bloodstream you produce, or you have, anti-A antibody. If you're blood type AB, since your red blood cells have both surface molecules A and B, you produce none of the antibodies, because the antibodies would attack your own blood cells. Okay? If you're blood type O, you produce neither, you have neither of the surface molecules, so your body produces both antibodies, A and B. Okay? So that's the natural condition. Natural situation, what somebody would be if they're blood type A, B, AB, or blood type O. So the problems occur when a particular blood type is mixed with a blood that has antibodies that would attack the naturally occurring surface molecule on a blood cell of a patient. So if there's a patient that has blood type A, they have molecule A on their red blood cells. Those red blood cells would be attacked by antibody B. I'm sorry, they would be attacked by antibody A, right? Anti-A antibody would attack molecule A. So notice the problem over here, which we will continue in the next section.